Hello, good afternoon, and welcome to Chemspec Digital. I have the pleasure to introduce the next presentation in our RSC lecture theatre. But before I do want to point out that we are recording this session and making it available to view on demand at a later time on the platform. And we are also inviting attendees to write questions into the chat box, and we will ask the presenter the questions at the end of the session. But now, without further ado, I will hand over to David Palmer of Micropore Technologies, who will be presenting membrane emulsification from lab to manufacturing monodispersity at scale. Over to you, David. Thank you.
Good afternoon, everybody. My name is David Palmer, Technical Support Manager for Micropore Technologies. I'm going to be covering membrane emulsification generally, and then a couple of case studies using the membranes in a slightly different manner. So a little bit about the company, if I can get my slides to change. There we go. Micropore is a spin-off from Loughborough University here in the UK. And we relatively recently, relocated to the northeast of England, up at the Wilton Centre, a state-of-the-art facility in the northeast. This houses our headquarters office, laboratory and engineering facilities. We've also got offices in the US and have recently opened an office in India. Now, as a spin-off from an academic institution, the technology for a long time remained a little bit of an academic hobby. It was great for university students to write the PhDs and get some nice small-scale lab samples to take pictures of for publication. It's only really in recent years that we've solved some of the problems and the challenges of scale-up, which I'm going to talk to you about today. So we have a technology platform based on membranes. You can see a micrograph in the bottom of the slide there. It's a sheet of 316 stainless steel with laser drilled pores of a well-defined diameter and distance between the pores that we call pitch, and also geometry. So it's using the control over this laser drilling process really allows us to have our patented membrane emulsification technology as a scalable system. Now, this allows us to manufacture monodispersed emulsions, which you can then turn into microparticles, microspheres, or in some other applications, nanoscale particles or crystals. So basically, we can target the droplet or particle size that you want with high yields, less waste, and improved repeatability and consistency generally. Because of the way the two fluids are introduced to each other, we reduce the chance of getting inversions when we're making emulsions, which is where your oil in water emulsion might flip to a water in oil emulsion, meaning that you have to shut down your manufacturing, clean out your tank, you generate a lot of waste material, lose a lot of production time. So by introducing the two fluids together through the membrane, we can dramatically improve the processing. So a bit of terminology, which is probably commonly known to most people, but just want to make sure I define it. We talk about injection of the dispersed phase through the membrane pore into the continuous phase. So that's basically forming a droplet of oil in a continuous phase of water, or a droplet of water in a continuous phase of oil. Or indeed, we can do much more complex multiple emulsions, whether that's oil in water in oil or water in oil in water. And for the water in oil type systems, we do have to apply a hydrophobic coating to the membrane, because how the dispersed phase wets over the surface of the membrane is very important to the success of your emulsification. Basically, as you inject the dispersed phase through the pore, you get a droplet forming on the surface of the membrane. We then use shear forces to deform and detach the droplets from the pore, and the result are droplets with a very well-controlled uniform size. So there's a lot of ways to traditionally make emulsions, especially at large scale. They tend to be things like rotor-stator homogenizers, high-pressure homogenizers or ultrasonic systems, or high-speed stirred tank systems. Now, these all apply a lot of energy. They're somewhat chaotic in that you have to monitor where you are in terms of particle size at any particular point in time. So they have to be monitored and measured to avoid being either oversized or undersized. It's a dynamic process. But inevitably, they will give you a wide size distribution. There are also other ways to make droplets. There are systems like microfluidics, where you have very, very good control over droplet formation. But typically, these are very, very low volume. So you're talking maybe grams an hour, grams a day. You know, you're working with microliters scale. So it's good to prove the principle, but then how do you scale up? Some microfluidics use very high pressure. 30,000 PSI, I heard on a recent webinar. So again, if you're working with very sensitive materials, they may not be suitable. Homogenizers obviously are very good at high volume applications, but use very high energy, wide size distributions, and can be quite high maintenance. They're also very loud. We fall somewhere in the middle. So we have solved many of the problems of using membrane emulsification at high volumes. It's a very low energy system. Basically, you're running two pulseless pumps, whether that's syringe pumps or gear pumps. We can target a very narrow particle size distribution, and they're very low maintenance. You press start on two pumps, you can walk away. So just comparing a typical system made by homogenization. This is an oil and water system where we've targeted 20 micron average droplet size. You can see you've got a very broad array of different droplet sizes. These will all have different volumes, therefore different buoyancies. So if you left this as a standard emulsion, you're likely to get creaming or sedimentation. It's an inherently unstable system. You're probably going to need a lot of surfactant or emulsifier to keep it stable. If you compare this with membrane emulsification, it's clearly very different. Every droplet is pretty much exactly the same size, again, targeting an average of 20 microns. And if you were to turn these emulsions into something like a microparticle or microcapsule, on the left, you're going to have a wide array of different mechanical strengths when you form the particle or the microcapsule. The large droplets are going to be very mechanically fragile. The smaller droplets might not release at all. So potentially you could have spent quite a lot of time and money encapsulating an expensive active, only to find out that it breaks when you don't want it to, or it doesn't break at all, and you've basically wasted a lot of time and effort. With membrane emulsification, everything is much more predictable, controlled, and repeatable. So as a spin-off from a university, we do a lot of mathematical modeling to look at how we can influence different chemistries, different particles, different processes. And part of that is through membrane geometry. We've got interchangeable membranes with different pore sizes. So the pore diameter is a key parameter. Typically, the rule of thumb is that a droplet formed from a pore will be between 3 and 10 times the pore diameter. We also have to consider the pore pitch, so how far apart the pores are from each other. So for example, if you're trying to make a 100 micron droplet and you've got 50 micron pitch, then you're going to generate droplets too close to each other. They're going to coalesce, and it's going to skew your particle size distribution. Equally, pore geometry, if you have two pores directly in front of each other or behind each other, and you have two droplets coming off from those pores at the same time, they can collide and coalesce, again, skewing your particle size distribution. So by offsetting the way the pores are arranged, we can minimize these coalescence events. So the main ways of influencing the particle size is through the injection rate, so how quickly a droplet grows on the surface of the pore, balanced against the shear stress we apply across the surface of the membrane. In most cases now, we use cross-flow membrane emulsification, by which I mean we use the flow of the continuous phase to provide the shear to deform and detach the droplets from the surface of the membrane. There are other approaches. Historically, we've used some quite mechanically complicated ways to apply the shear. through oscillation, whether vertical or horizontal. These worked at small scale, but proved very difficult to scale up. The other thing you have to know, obviously, is some knowledge about your chemistry, such as the viscosity, the interfacial tension, the type and concentration of emulsifier or surfactant you're working with. But once you consider all of those things, we can basically target a particle size pretty easily. So you can see in the pictures on the bottom here, these are all made using a single membrane. By changing the injection rate and the rate of shear, you can basically dial in to any specific emulsion size that you want, whether that's for making microparticles, emulsions, or doing emulsion polymerization. You can very carefully control the relative ratios of your dispersed phase and your continuous phase. It's a very gentle process due to the membrane design. So it's very good for processing things like sensitive biological materials. We've run studies where we have basically taken emulsion containing yeast particles and put them through a membrane, and 90% of the yeast remain viable through fluorescence microscopy, excuse me. And we've done the same system through a rotostator homogenizer and found that 45 to 50% of them are viable. So it is a gentle process if you're working with sensitive raw materials. So these are all cross-flow membrane emulsification devices, and really I just want to highlight the top line of the table here. For a long time, the maximum capacity for this kind of technology was lab scale 10 or 20 mil batches, or 10 or 20 mil injections into 100 odd mil of continuous phase. So very small batch scale. Now we've introduced a small lab scale device called the XF Mini. This has been explicitly designed for pharmaceuticals with the lowest hold-up volume that we could manage, because if you're working with very expensive APIs, you don't want to have to waste even a gram if it's costing you £1,000 a gram. So we've tried to design it to be as economic as possible, and yet capable to go up to 20 litres an hour with a device that will sit in the palm of your hand. Equally, the XF1 will sit in the palm of your hand, 6 litres up to about 200 litres an hour. And then finally, we have the XFN, which is a modular system, which allows us to scale up in a controlled linear way. In this case, it's configured as an XF7, allowing us to produce in the region of 1.5 tons an hour. You can't hold this in the palm of one hand because it's too heavy, but you can easily hold it in 2 hands. So the equipment footprint is very small, and they just need 2 pumps to run. So once you've made these emulsions, what can you do with them? Well, you can do all sorts of interesting chemistry with them. So starting on the top right, we've recently been doing some work with liposomes and lipid nanoparticles. We've been doing some interesting work with API crystallization, and these are non-emulsion based approaches, which I will cover in the next few slides. We can also cover hydrogels, materials such as alginates, chitosan, carrageenan, agar. These can be considered to be diffusive systems. It's really like a fishing net of polymer strands holding your active inside the particle. So it could be used for delayed release in agriculture or drug release, or an air freshener block is essentially a hydrogel with a fragrance in it. It just diffuses out slowly over time. And depending on the hydrogel and the density and the concentration that you use, you can control the release of that to some extent. Then we have the more traditional core shell microcapsules. These are much more binary in terms of release, as in there's a physical wall or shell around a droplet, and it's either broken or it isn't. But you have some very flexible and versatile chemistries there, which again, I'll highlight a few examples of. We can also produce mesoporous silica particles where we can control the porosity of the particle by choice of surfactant and concentration. This then allows you to diffuse actives into the silica particles, where you then have the option to potentially post-coat them with something like a pH responsive polymer. meaning you have a combination between a core shell capsule and a hydrogel, because basically once you dissolve the pH sensitive polymer, you can then have a diffusive release. We're also looking at some opportunities in continuous flow organic chemistry, which is quite interesting. And we do quite a lot in the pharma space at the moment with biopolymer microspheres. So these tend to be biodegradable drug delivery systems, materials like PLGA, so it's polylactic glycolic acid, or polycaprolactone, or polylactic acid. So PLGA is a good example. It's very commonly used. It's FDA approved. You can basically tailor the polymer backbone with different end groups, which allow you to control the rate of biodegradability. You can also do that by particle size and other in-process adjustments. And these are typically used for things like injectable drug delivery. You'd encapsulate your active, whether it's an anti-cancer or a monoclonal antibody or some other therapeutic material. You can inject it at the right part of the body where the particles will lodge and will basically break down over time, act as a depot of the API, and will release in a controlled manner, consistently applying the therapeutic as required. And there's lots of these applications being used commercially today. We have somewhere in the region of 11 in preclinical and a few in clinical at the moment. So why is that important? Why is monodispersity important for injectables? One reason is if you have very broad particle size distribution, you have to use a very big needle because you don't want it to block halfway through the application of the dose. Your patient doesn't know how much dose has been given. Large needles cause significant discomfort, and yeah, they will probably have to go to a clinic to have the injection applied. If you have very monodisperse particles, you can basically use a lower gauge needle, much less painful for the clients, and they could probably self-administer at home. Also, each particle has the same amount of drug in it, and it's going to break down at exactly the same rate based on its size. So on the left, you can see a pretty traditional oil and water emulsion using PLGA. But on the right, you can see that we can design some more complex systems. This is a water and oil and water particle. This allows you to potentially work with hydrophobic and hydrophilic actives in a single particle. So reducing the number of doses that a patient might have to have. You can also see the influence of polymer concentration on and encapsulation efficiency, basically. We also do some more industrial chemistry. So we can do interfacial polymerization type systems, whether that's polyurea, polyurethane, or polyamide. And you can either have your monomer, one in the dispersed phase, one in the continuous phase, reacting at the interface. You can have both monomers in the dispersed phase, but depositing the polymer at the interface. There are a number of different systems initiated in different ways, but again, a very versatile platform for controlling the size and therefore the relative volumes of monomer in your particle, and therefore residual monomer in your particle. And we're actually applying this chemistry as part of the European funded project called SmartInks, which is looking at self-healing approaches in cementitious systems, so self-healing concrete, essentially. We're involved in a project basically involving 15 researchers, a multi-industry partner and European university research project, which you can learn more about online if you're interested. Then we can also process some of the more classic chemistry. So complex classivation was one of the first real industrialized encapsulation processes. It was developed in the 1950s and really used mainly in carbonless copy paper. So you basically form your emulsion. You would have a couple of biopolymers dissolved in the continuous phase. For example, an amphoteric gelatin, a anionic material like gum arabic or CMC. The gelatin is amphoteric. So if you drop the pH of the system, the gelatin becomes cationic, interacts with the negatively charged polymer and forms this polymer-rich coacivate phase within the aqueous phase. And by controlling the temperature, you can control the deposition of this material around the oil droplets and basically form a wall or a shell around the oil droplets, which you can then chemically cross-link. And you can use this from anything from flavor and fragrance delivery. We've used these as damage indicators in composite systems. They're food grade, so you can use them to deliver actives through the low pH of the stomach, but will break down in the lower intestine, for example. So again, a really versatile encapsulation platform. So one of the case studies I wanted to talk about today was LNPs, lipid nanoparticles, and why scale is important. There's been a lot of interest in LNPs recently, partly because of the development of the mRNA vaccines. A couple of the COVID vaccines use this technology. You can't just inject mRNA into the body. It gets degraded by some of the body's natural processes. So you basically have to smuggle it into the body using basically a lipid sort of covering for protection. And we found that we could make these materials using our membranes as a micro mixer, specifically using the ethanol injection method, where you have your lipid formulation mixed in with the ethanol. You then mix it in with the material to be encapsulated and any formulation aids that are required. But it's the important part of this is the mixing. If you have poorly controlled mixing, these phospholipid bilayers, they self-assemble into different structures. If you don't have good control of mixing, instead of unilamellar, as in single shelled LNPs, you can get multilamellar particles. And this is indicated by the size distribution. If the size is too large, you've probably not sure what you've actually made. So control of mixing is really important for making these things, especially at scale. So what we do is we take our cross-flow device and we inject the lipid and ethanol in as if it were our disperse phase in a standard emulsion, essentially. Ethanol is miscible in water, so it's not really emulsification at all. We then take our material to be encapsulated and consider that to be our continuous phase. As the two meet, you have your phospholipid bilayer with a hydrophobic end and a hydrophilic end. The hydrophobic end wants to get away from the aqueous environment. So you get these discs beginning to bend and then basically self-assemble into a sphere with your hydrophilic ends on the outside, your hydrophobic ends on the inside, and hopefully your material trapped in the middle. So we knew that we could make things this way. We didn't know quite what we had made. We are an engineering firm who does some chemistry. We're not experts in this area of biology in particular. So we commissioned this study with the University of Strathclyde, guided by Professor Yvonne Perry, and we used our aseptic stainless steel membranes to look at a number of different conditions. We wanted to be able to produce at a reasonable rate. So TFR here is total flow rate. We wanted, or we knew from literature, what target size we wanted, the polydispersity index, the PDI, again from literature, and we wanted a reasonable loading. Poly A is used here because mRNA is actually extremely expensive, as you might imagine. So we were using poly A here as a placeholder to develop knowledge about the system so that we could then go on and apply our learnings. So basically what we did was take a model formulation and we gave Strathclyde the device itself and a couple of pumps and basically taught them how to use it, and away they went. And in the first few runs, we did it incredibly well. We hit the target size, we hit the target PDI, we're monitoring the zeta potential, and we're getting very, very good encapsulation efficiencies. As this is about scaling up and understanding the manufacturing process, we wanted to see what levers we could pull, what effects different configurations and different flow rates have. So basically, again, taking a starter formulation, we looked at the total flow rate. So as you increase the flow rate, you're basically increasing the shear or the flow of the continuous phase over the membrane, and therefore generating smaller particles. So yeah, we can manufacture slowly and get a particular size, or we can manufacture more quickly and control the size that way. We also wanted to understand the influence of the membrane, because obviously we're using micron-sized pores to generate nanometer-sized particles. So by looking at the difference between a 40, a 20, and a 10 micron membrane, obviously you can appreciate that it allows a different volume of the ethanol and lipid to be mixed at any one time. So again, we can see the influence of size there. We still maintain a good polydispersity index. As part of our process control, we can also control what happens inside the tubular membrane. So we can basically put something in there which will control the annulus height and therefore the velocity of the continuous phase. So the larger the annulus, the lower the viscosity. So unsurprisingly, if you narrow the annulus, you basically increase the velocity and therefore the shear, and it has a corresponding effect on the particle size. So then we actually bit the bullet and encapsulated some mRNA, and we did some pretty basic tests. And we were looking in particular at cytotoxicity, and pleased to report no toxicity was reported. We were looking at the cell uptake. Again, concentration dependent uptake is something we were very pleased to see. And then the expression of the mRNA, and again, pleased to see strong cell expression. Most importantly for me as a non-biologist, what we put in when we looked at it came out exactly the same. So we have not denatured or degraded the mRNA in any way, which is really important, especially when manufacturing at large scale. The second case study, which I will try and get through relatively quickly, is looking at API crystallization and using our membranes as micro mixers again. Basically, by taking a solvent with an API in it, if you apply that into a non-solvent, you can control the how the crystal forms, crystal size, crystal morphology, and these are important parameters in pharmaceuticals for bioavailability. So if you imagine our membranes, each pore is acting like a little microfluidic channel, and it's nucleation control, so we can make many small crystals at the same time. Again, we've done this using our AXF crossflow device, and what's happening here is the disperse phase is your solvent and your API. It's going into your anti-solvent continuous phase. You're getting micromixing at the pore and basically forming crystals of a controlled size and shape. Now, there's two devices on screen here. One is the AXF Mini, and next to it is its predecessor, like a heritage device, which was a dispersion cell. This is a very small scale batch process, which has now been superseded. But I put the image there because I'm going to report some data using the LDC on the next slide. So here we use the dispersion cell to look at a crystallization process. We then applied the same mathematical model as you saw earlier to look at the scale up on the AXF, and you can see that this particular API crystallizes extremely well at larger scale. Again, here we've replaced the LDC with the Mini. So this is our new lab scale device. You can see that it maps pretty well, again, from the Mini to the AXF. So this is from 60 mil up to potentially 200 litres an hour on the AXF. We've also looked at repeatability and reproducibility studies and are getting some quite good results. So really starting to sum up, we have 3 devices using membrane emulsification. Very versatile platform for making emulsions, microparticles, lipid nanoparticles, liposomes, but also some potential for reaction chemistry in continuous manufacturing. We've really finally solved the problems of scale up. We've removed a lot of the mechanical aspects like the stirred cell or the oscillating membranes. Now we purely focus on the cross-flow principle to produce our particles. So in summary, very, very simple operation, no moving parts, very easy to use, to clean, to sterilize, all 316 stainless steel. You can use one system for all of the examples I've given you today, from a range of 60 mil an hour up to about 1.5 tons an hour, using a very low shear, highly reproducible system and very gentle processing, in particular if you're using very sensitive biological materials. And that, I think, is allows me to conclude the presentation. Thank you everybody for your interest, and if you have any questions, I'll be more than happy to answer them.
Lovely. Thank you so much, David, for that. And the thank you is not only coming from me, but also from a member of the audience in saying that it's a very informative lecture, which I agree. And the question is there a difference, or what is the difference between microsieves and micropore membranes?
Nomenclature, basically. I mean, there are other people using membrane type structures, obviously for filtration. We actually started out as a filtration company ourselves, but found that there was actually more money to be made putting things through rather, as an emulsion rather than trying to filter things out. So microsieves can be used in some specific membrane emulsification processes. I think there's a Dutch company doing both applications.
Okay. Excellent. I hope that answers that question. The other questions was how many times can a membrane be used?
So as they're made of 316 stainless steel, they're all laser drilled. Unfortunately for us, they go on pretty much forever, unless you happen to jump up and down on them, which some people have managed to do. They're very, very robust. And this is one of the advantages of our approach over some of the others. There are some silicon wafer, or there's some glass membranes out there, which obviously have some challenges, especially if you're working in food or pharmaceuticals with very fragile membranes.
Okay, and the last question was: do they get blocked? Do the membranes get blocked?
If you're working with particulates, you have to be aware of the size and the aspect ratio of the particles you're working with to avoid blockage. So if they're long needle shapes and they will go through needle shape on, that's fine. If they go across the pore, then you've got a chance of blockage. We can apply pressure gauges to all of our systems, so you can monitor the open area of the membrane and therefore have confidence that every pore is operating as expected. So blockage is rare, but it can happen, yeah.
Brilliant, thank you. I think that, in fact, is concludes our question and answer session. I'm wondering whether there's anything else coming in. Then I think do reach out to David. He's got his contact details on the screen at the moment. I would like to say thank you so much for your presentation, David, and for making time to come on board. I know that's a busy time for you at the moment. I'd like to thank everybody for watching and for attending Chemspec Digital. We hope that you still have some meetings scheduled for the rest of the afternoon, and see you all next year in Frankfurt. Thank you. Bye-bye.
Thank you. Goodbye.